Oh, Daniel, you're muted.
Thank you. Welcome everyone to the DFACC Connect Science. My name is Daniel Haber. I'm the director of the Cancer Center at Mass General Hospital. And our speaker today is Rob Manguso. Rob is an assistant professor at Harvard Medical School and the Mass General Cancer Center and a scientific group leader at the Broad Institute of Harvard and MIT. He received his PhD with Nick Haining at Harvard and at the Broad and has achieved fame and success in looking at in vivo CRISPR screens, looking at modifiers of sensitivity to immune checkpoint inhibition. He directs a program called TIDE at the Broad called Tumor Immunity Discovery Engine. The title of his talk today is In Vivo CRISPR Screens Reveal the Landscape of Immune Evasion Pathways Across Cancer. And before turning it over to Rob, I would ask if you have questions during the talk, please type them into the Q&A, not the chat box, but the Q&A. And at the end of the talk, then I'll call on you and we'll have the questions answered. Thanks very much, Rob.
Thank you for the kind introduction, Daniel. And thank you everybody for being here today to listen to me speak about my lab's work. Today, I'm going to tell you about work we're doing to use in vivo screens to identify new immune evasion pathways. This is my disclosure slide. So the work in my lab, sorry, is this Zoom thing visible to you, or is it just me that sees this?
Looks good.
We do see it, but only when the mouse goes up there.
Oh, okay.
But we are seeing it now, actually. Okay, it disappeared.
Okay, that's great. So the work in my lab is motivated by the observation that immunotherapy with checkpoint blockade for cancer has been remarkably successful for many patients with difficult to treat malignancies like advanced melanoma, shown here on the left, and non-small cell lung cancer, shown here on the right. But the major observation that is motivating the work is that it's not effective for everyone. So particularly here in the non-small cell lung cancer survival curve data, you can see that there are a couple of different classes of patients that are resistant to immune checkpoint blockade. One that appear at the top of the chart here to really derive almost no benefit from immunotherapy compared to chemotherapy. And this is what we've started to call intrinsic resistance or primary resistance to immunotherapy. And then there's a second group of patients here that do appear to derive some benefit from anti-PD-1 blockade with pembrolizumab compared to chemotherapy, but they eventually do succumb to their disease. And this is what we're calling acquired resistance. In general, we need to understand the immune evasion and resistance pathways that are leading to this type of resistance and come up with new approaches that would be able to use immunotherapy for a greater number of patients. Sorry, I have to move this thing again because I can see it. Okay. Actually, I think I can make it disappear. There we go. Okay. So, the other problem is that we're talking about when we're talking about non-small cell lung cancer and melanoma, cancers with a relatively high mutational burden that are among the most responsive to immunotherapy. So this plot from this nice review in the New England Journal in 2017 shows the average response rate to PD-1 or PD-L1 blockade across many different types of cancer, graphed against the mean number of coding mutations per megabase in that particular tumor type. And you can see that there is a trend here where tumors with a higher average mutational burden generally also have a higher average response rate to PD-1 or PD-L1 blockade. Melanoma is up here almost right on that curve, one of the higher end. And then there's mismatch repair deficient tumors, which are also well appreciated to respond very well to immunotherapy. And then there's a large group of tumors here with a relatively low mutational burden and also, on average, less than a 25 or 20% response rate to immunotherapy. And it is for this group of patients who are primarily not benefiting from immunotherapy that we need new therapies to increase the ICB response. And these could be combinations with existing drugs like PD-1 or PD-L1. The clinical development landscape for combinations with PD-1 or PD-L1 has exploded in the last several years because of exactly this challenge. So you can see here, these are graphs that represent the number of combination trials going on with PD-1 or PD-L1 blockers. So each circle is a target, and the size of the circle roughly corresponds to the number of ongoing trials with PD-1 or PD-L1 blockade for that target. You can see that from 2017 to 2020, there was just a huge explosion. There's a massive number of trials in the center here with a few major targets, and then hundreds of other trials ongoing with other targets that are putting a strain on our ability to recruit patients actually for the trials. But nevertheless, this is extremely important work because there have been notable successes in this space. So many of you who are following the immunotherapy field are probably familiar with the fact that several years ago it was shown that actually the first immune checkpoint blockade drug, ipilimumab, which blocks CTLA-4, which is expressed on T cells, combined with nivolumab, improved the response to immune checkpoint blockade over nivolumab alone. And more recently, there's been another notable success, relatlimab, which is an anti-LAG3 monoclonal antibody. LAG3 is another immune checkpoint expressed on T cells. And compared to nivolumab alone, the relatlimab-nivolumab combination did improve the response. So there are important and notable successes here, but it took a huge amount of clinical development activity in order to come up with these successes. And I believe we need more systematic methods for discovery and preclinical assessment of new targets, and that genetic screens could offer a way to both identify new targets and pathways, but also provide information about their relative priority and how strong an effect size we might expect from trying them with any given immunotherapy as a baseline. So I'm going to describe our work today using genetic screens for exactly that purpose. But first, I'm going to familiarize you with the model systems that we're working in. So we rely pretty heavily on these mouse models of tumor immunity. These are cell lines that can be grown and manipulated in a dish. There are things like B16 melanoma, which many of you might be familiar with. These are cell lines that are derived from either spontaneously occurring tumors in mice or genetically engineered mouse models or carcinogen-induced mouse models. And they can be grown in a dish and then implanted into wild-type, totally immunocompetent mice, where over the course of a couple of weeks, the tumor will grow until the mouse succumbs to the disease. But because these are immunocompetent mice, they can also be treated with the preclinical versions of drugs like PD-1 or PD-L1 blockade. And this provides us a model system that we can use to study how these therapies work, and indeed to maybe develop discovery screens that could identify targets that work with PD-1 or PD-L1. So several years ago, we devised an experimental strategy to do just that. We engineered some of these mouse transplantable tumor models to express Cas9, and then delivered guide libraries to them and selected them in vitro, just like you would do for any screen in vitro for any purpose. But then the difference between what we're doing and other dependency screens is that we're implanting these library transduced cancer cells into mice. And we implant them into groups of mice that would allow us to identify guide RNAs targeting genes that are having an effect on the fitness of tumor cells in response to selective pressure from the immune system specifically. So we always implant the tumors into a group of immunodeficient mice. Then we implant them into wild-type mice that we don't treat with anything, and then wild-type mice that we treat with our immunotherapy of choice. And when we harvest the tumors and sequence the guide RNA barcodes from tumor genomic DNA, we can compare the data that we get on barcode abundance from the immunotherapy treated mice or the wild-type untreated mice back to immunodeficient mice in order to find genes and guide RNAs that are increasing the fitness of tumor cells in response to immunotherapy. So these things would be mimicking resistance mechanisms, or genes that, when deleted, increase the sensitivity of tumors to immunotherapy. These would be what we call immune evasion genes and would represent new immunotherapy targets. And I'm going to focus today mostly on these genes that are becoming that are depleted in our screens and are increasing the sensitivity of tumor cells to immunotherapy, because they're the most interesting. We think they represent potentially new druggable targets. So several years ago, after doing the first proof of concept screens using this experimental system, we began a project at the Broad called TIDE, which stands for Tumor Immunotherapy Target Discovery Engine. And the goal of this project was to do this kind of experiment at the largest scale possible. So in the most mouse tumor transplantable models that we could get our hands on and get to work in the screens, and at the largest screening scale possible, which it turns out we were able to do it at genome scale. So we ended up being able to do the experiment at genome scale for 8 different mouse transplantable models. We tried to hit several of the models that are commonly used in the field, like B16 and MC38, in addition to several others that represent a few different tissue types, and then subgenome screens for a subset of those. So it's really a nice data set of immunotherapy dependencies across many of these mouse cancer models. In addition to the screening data, we also generated some characterization data on the lines. So we have transcriptomic data at resting state for each of the lines. And actually, we generated transcriptomic data for each of the cancer cells' response to a few different inflammatory cytokines that are present in the tumor microenvironment, like interferon gamma, type 1 interferon, and TNF alpha. And then finally, we actually have single-cell RNA sequencing data of total CD45 positive cells from the tumor microenvironment of each of these models, with and without the immunotherapy that we're using to do the screens. So we have an idea also for several of them of what's going on in the tumor microenvironment with regard to changes in the infiltration or state of tumor infiltrating lymphocytes. So just one aside before I continue on to talk about some of the screening data. In order to accomplish these screens, we actually had to develop some new technology, which we published in a paper in Immunity last year. So for many groups that have tried to do these kinds of experiments, it's a well-appreciated problem that the expression of Cas9 and other proteins derived from the vectors that are normally used to do these kinds of experiments, like resistance genes, are potentially problematic immunogens and can cause the rejection of transplantable cancer models in immunocompetent mice. So in our paper, we show this pretty convincingly, that in a couple of the commonly used models, the CT26 colon cancer model and this BRAF-PTEN melanoma, that even in the absence of any immunotherapy treatment, tumors transduced with Cas9 vector and this guide RNA vector are just rejected by the immune system. And then in another model, the KPC pancreatic cancer model, they would grow in wild-type mice, but their response to immunotherapy is dramatically enhanced by the expression of these vectors in a way that seems to be not physiological. So we developed a solution for this problem that would allow us to do the screens, and I'll describe that solution here. We developed a vector system that we call SCAR, or selective CRISPR antigen removal, and this system eliminates all the expressed antigens using the CRE-LOC system. So the first vector is this pSCAR-Cas9 vector. It's actually quite a useful Cas9 vector for many reasons. It has Cas9 2A linked to blastocidin resistance, which actually generally improves the genome editing rate. And it also has a visible reporter, GFP. It has a LOX-P site encoded in the 3' LTR region, and this duplicates upon integration into the host genome, so the entire vector insertion is floxed. We have a guide RNA that we call pSCAR guide RNA vector, where the entire, so we have pyromycin resistance physically linked to a fluorescent protein called MK2. We actually have a few different versions of this. And this entire region of expressed protein is flanked by LOX sites that are incompatible with LOX-P, but the U6 guide RNA region is not floxed. And then we have a vector that we package into non-integrating lentivirus that expresses CRE transiently. So after expression of CRE, you get a recombination of the LOX sites. It leaves only a scar in the genome from this 3' LTR here for the Cas9 vector, and it eliminates all the expressed regions of the guide RNA vector except for the U6 guide region, which you need to read out the results of a screen. And we showed with a couple different tumor models, but I'll just highlight one here, that this vector system completely eliminates the problem of the tumors being highly immunogenic in vivo. So unmodified KPC pancreatic cancer cell growth in mice is shown in gray. If you express all of these vectors but don't add CRE, they express a lot of antigens and they're pretty rapidly rejected by the immune system. But if you add the transient CRE delivery, that completely reverses the immune rejection in red, and the tumors grow at a normal rate. So all of this is described in our paper, and all the vectors are actually available on AdGene for the entire community to use. So using this technology, we were able to develop immunotherapy treatment regimens for each of the models that we were going to perform a screen in that did apply a decent amount of immune-based selective pressure to each cancer model. So for actually most of them, we needed to use a combination of anti-PD-1 and anti-CTLA-4 because they're quite refractory to immunotherapy. But for a few of them, we were able to use PD-1 alone. And we were able to perform screens in each of these models that generated a really beautiful landscape of different immunotherapy dependencies in each model. For all 8 of them that we were able to perform genome or subgenome scale screens in, we were able to identify statistically significant hits for genes that would increase the sensitivity to immunotherapy when deleted. Interestingly, for some of them, we were not able to generate statistically significant resistance-causing hits. And we don't think this is a problem with the screen performance. This is actually potentially a result of the fact that the models may not, because of the immune response generated, there may not be any single gene knockout phenotypes that can render them completely immunotherapy resistant. But we do find among the resistance mechanisms some nice positive controls, like NLRC5 here, which is a transcriptional regulator of MHC class 1, P10, which has been described to cause immunotherapy resistance when deleted, and caspase-8, which has also been described to cause immunotherapy resistance. And in addition to just finding a lot of hits from the screens, we know that we can validate these things too. So for a few of the hits that we got in our B16 screen, we were able to show that they have the expected phenotype in single gene knockout validation studies. So for the gene CCAR1 here, which we predicted to cause resistance to immunotherapy when lost from the screening data, it does exactly that in an individual validation study. And for RNF31 or calreticulin, both of which were predicted to increase the sensitivity of B16 melanoma tumors to immunotherapy, we can see that we can validate those results as well. So the in vivo screens can not only identify immune-related dependencies, but these dependencies are also largely validating to increase sensitivity in the direction that we expected. So one of the interesting things about the data set is that actually most of the individual hits from each model are unique to a single model, but that doesn't mean that nothing is shared. So there are a few different ways to look at what commonly is coming out of the screens. One of them is just by looking at the shared depleted hits. So these are, again, sensitizing hits from each screen. And we're comparing our immunotherapy treated condition to the immunodeficient mouse condition, which for these screens was actually NSG mice. And this is a representation of genes that have scored in 2, 3, 4, 5, 6, and 7 models. There were no genes that actually scored in all 8 of the screens. And actually the only gene shared across all 7 models was the gene H2T23, which encodes the mouse homologue of HLA-E, a non-classical MHC1 that we're going to talk more about later in the talk. Another way to look at this data is to aggregate the entire data set into one large screening data set and plot it as a volcano plot. So you can see here that when you do that, the gene H2T23 comes off as off to the left as the most commonly found depleted target among all the screens. So, one lesson that we learned right away from looking at all the data is that the mouse homologue of HLA-E is probably the most common and important single immune evasion gene that we discover in most of the models. And this is held true for some other screens that we've done now in additional models that I haven't shown here. Another way to try to identify the most important pathways that are scoring commonly across the models is to create gene sets of the significantly depleted immunosensitizing hits, and then look at the aggregate screen score for those gene sets in each of the different models. with here the size and the color of the dot representing the adjusted p-value and the strength of the recovery of the hits in an individual screen. And when we do this and plot it in a heat map, you can see a couple clusters that come out here. One relating to MHC1 antigen processing and presentation genes. So these are genes that are involved in the peptide loading complex that's responsible for loading antigens onto MHC1 and some of the MHC1 heavy chains and B2M. And then several genes that are necessary for sensing and signaling for interferon. So the interferon gamma receptors, and in some cases, the type 1 interferon receptors, and then several members of the JAK-STAT pathway that are necessary for signaling in the interferon cascade, and occasionally also PDPN2, which is a negative regulator of interferon sensing. And when you take all of these statistically significant hits and you perform a string protein-protein interaction analysis on them, it results in a network that looks like this. And you can see that there are two major nodes in this network, one corresponding to the genes in the interferon sensing and signaling pathway, and one corresponding to MHC1 antigen processing and presentation genes. Most notably among them is H2T23 here, which again encodes for the mouse homolog of HLA-E. So we decided to focus more on the observation of the interferon sensing pathway because it was such a strong observation in our screens, and because this is a bit of a counterintuitive result, but I'll get more into that for why we think it's counterintuitive in a minute. So all of the screens that I've described so far are performed in vivo. So the selective pressure is potentially being applied by many different immune populations. But screens like this have been done using immune cell co-culture assays as well. Many different groups have published screens where T cells and tumor cells are mixed in a dish to find the genes that are necessary for T cell killing. So we had an opportunity to compare our data generated in vivo to data generated in vitro with specific selective pressure from T cells. The specific data set we chose was a data set from Jason Moffat's group that was published in Nature in 2020, because he had performed a similar analysis of screens from 6 different cancer cell lines, some of which are overlapping with the screens we performed. So it was a pretty decent comparison between the two data sets. And in their paper, they describe the interferon and MHC1 sensing genes as necessary for T cell killing in vitro. Indeed, when you plot our in vivo screening data against their screening data, so here on the y-axis is our aggregated in vivo screening data set with depleted genes following on the bottom and enriched genes at the top, and the same for their data set with depleted genes on the left and enriched genes on the right. You can see that there are many genes that agree between the two data sets that are captured in the top right and bottom left quadrant that are sensitizing or resistance causing in both data sets. But there's this large cluster of genes in the bottom right that are discrepant between the two screens. And these have some of the largest effect sizes in our data set and their data set. So the genes in the MHC1 antigen processing and presentation pathway and the interferon sensing pathway are causing resistance in the Moffitt Labs in vitro T cell screens, but increasing sensitivity in our in vivo screens, which is another reason we were very interested in this and wanted to follow up more. So interferon can have opposing effects in different tumor models, and we know this and actually validated it using models that we have in-house and perform the screens in as well. So here in the B16 melanoma, we can delete JAK1 and show that that causes resistance to immunotherapy compared to control here, where the control tumors have a nice transient response to this tumor cell vaccination and PD-1 blockade that is abrogated in JAK1 knockout tumors. However, the exact opposite thing happens when we do that experiment with this KRAS P53 pancreatic cancer model, where JAK1 deletion actually increases their sensitivity to PD-1 blockade. So, we know that this result validates in some of the models that interferon sensing genes had scored in our screens. So this is reminiscent of conflicting observations about interferon that have been observed in the literature. So there are both pro and anti-tumor responses, roles for interferon that have been described. So interferon signaling is important for immunosurveillance, and we've known that from early work by Bob Schreiber and others. Interferon is necessary for early tumor immunosurveillance. And more recently, several groups have described mutations in interferon sensing that are associated with resistance to immune checkpoint blockade in melanoma. So this notable paper here from Tony Rubis's group showing that in a patient with acquired resistance to immunotherapy with pembrolizumab, there was a mutation in JAK1 that emerged only post-treatment. But there's also a decent literature of potentially immune inhibitory effects of interferon. And this, there are a few different groups that have published on this, but largely Andy Min's group at Penn has published several papers on this. And Tom Gajewski's group has also published on this observation. So it's also known that in some of the models, deletion of the interferon sensing pathway could also enhance responses to immunotherapy. And that's what Andy Min's group is showing here in this plot, where deletion of the interferon alpha or gamma receptor genes would cause actually an increased response to immune checkpoint blockade. So several different mechanisms have been reported for what might be responsible for this effect. All of them are a bit conflicting, and we couldn't identify in our data set any evidence that those were the major mechanisms at play. And because the interferon pathway was scoring so strongly and recurrently as immunosensitizing when lost in our data set, we wanted to explore this more and understand why that's happening. But before doing that, we also wanted to know whether or not that this was a worthwhile endeavor. Is there clinical evidence that this is actually happening? So we looked for evidence that interferon would have the same opposing outcomes in patient data that we are observing in our mouse preclinical models. So the way we did that was taking published cohorts of patients being treated with immune checkpoint blockade. Here is data from David Liu, Ellie Van Allen's group in 2019. They published a nice, well-annotated melanoma cohort, and we scored each patient for which there was both transcriptomic data and response data as either high or low interferon sensing by the aggregate score of their interferon response genes, the hallmark interferon response signature. And patients with a high interferon score were predicted, did better in response to pembrolizumab than patients with a low interferon score. However, when we do the exact same analysis using a cohort of patients with RCC that was published by David Braun in 2020, we get exactly the opposite result, where patients with a high interferon score are doing worse in response to anti-PD-1 than patients with a low interferon score. So very reminiscent of the opposing data that we generated using the mouse models as well. So we wanted to understand the mechanism that might be responsible for this. And we, in order to give a little background into how we're thinking about this, I want to just give everyone a reminder of what interferon signaling and sensing actually does. So interferon gamma is an important inflammatory cytokine that's produced by effector cells like CD8 T cells, CD4 T cells, and NK cells. And sensing of interferon by tumor cells or any cells in the immune system causes a signaling cascade that signals through the JAK-STAT pathway and eventually causes the upregulation of a suite of hundreds of genes that we call interferon stimulated genes or ISGs. And for all of the mouse cancer models that we were using, we actually know what those ISGs are because we did transcriptional profiling of unstimulated and interferon gamma stimulated cancer cells for each model that we did screens in. So we know what their immunotherapy, we know what their interferon response is. So we reasoned that if any of the, if the immune inhibitory effects of interferon are caused by the upregulation of interferon stimulated genes that are having an immune inhibitory effect on anti-tumor immunity, then we should be able to discover those because we have both genome-wide screening data and the transcriptomic data. So we plotted that data against each other. And here on the y-axis, we're showing the in vivo immunotherapy dependencies that we generated from our genome-wide screens versus the interferon inducibility of that gene for every gene in the genome. And when we do that, you can see that there's a PD-L1 is highly induced by interferon, but doesn't actually score very strongly in our screens. And there's a group of genes here to the bottom right that are very strong immunotherapy dependencies in our screen and are also highly induced by interferon. And these are largely the same MHC1 antigen processing and presentation genes and a few members of the interferon sensing pathway itself that are recovered as the strongest hits in our screen, including, again, this gene H2T23. So this data is strongly suggesting that actually there's a clear mechanistic link between the immune inhibitory effect of interferon and the upregulation of these MHC1 antigen processing and presentation genes. Now, this is, again, a bit counterintuitive because we usually think of these genes as essential for CD8 T cell recognition of tumor, but I want to note that H2T23, this non-classical MHC1 gene here, is primarily an inhibitor of T cell function. So given the link that emerged from just doing an analysis of our screening and transcriptomic data, we wanted to validate whether this link appeared to be functionally true using this competition assay setup. So the way the experiment works that I'm going to describe here is we're doing a competition assay between control guide RNA tumor cells and tumor cells with a guide RNA targeting either interferon gamma receptor 1 or JAK1. So the competition is essentially mimicking the screen we did. We expect that when we treat the tumors with immunotherapy, guide RNAs targeting the interferon gamma receptor or JAK1 will be depleted relative to the control guide RNAs. But we're also going to do that same experiment on the background of tumor cells that lack TAP1. These tumor cells, TAP1 is essential for antigen processing and presentation. It's the transporter that allows peptides to move into the endoplasmic reticulum and be presented on MHC1. So a TAP deficiency should impair MHC1 presentation. And if MHC1 presentation is necessary for the immune inhibitory effect of interferon, then we would expect to lose that effect on a TAP deficient background. So when we do this competition assay, we get exactly that result, that there is a genetic interaction between MHC1 and the interferon pathway. So in the control background condition in gray, we see a depletion of guide RNAs targeting interferon gamma receptor 1 or JAK1 in both the KPC and CT26 tumor models. But in purple, on the background of TAP1 deficient cells, we see that when tumors can't present or have an impaired presentation of MHC1, that loss of JAK1 guide RNAs is reversed or substantially decreased, suggesting that the presentation of MHC1 is necessary for the immune inhibitory effect of interferon. Another way to look at this is actually to do a similar genetic interaction study in the patient data. And we can do this because it just so happens to be the case that the region containing TAP is actually frequently lost in cancer. So the TAP locus is on chromosome 6p21. This contains several antigen processing and presentation genes, but the major deletion bands are right around TAP1, TAP2, TAP-BP, which encodes TAP-ASIN, and a couple of proteasome subunits. And 6p21.3 is actually lost fairly frequently in many different types of cancer. But importantly, it's actually not, in most of those types of cancer, significantly associated with better or worse survival. So the analysis that I'm about to show is not really confounded by that effect. So the data that I showed you before from the RCC cohort, where I showed that a high interferon gene expression signature predicted poor response to immunotherapy, was actually in patients with 6p21.3 disomy. But if we do the same analysis in patients with 6p21.3 deletion, we actually lose that effect. So it also appears to be the case that in the patient data, the immune inhibitory effects of interferon also appear to depend on the tumor cells having optimal expression of MHC1. So what could be causing this effect? We wanted to understand mechanistically what's actually going on here at the cellular level. And we had a clear hypothesis, which is related to the biology of NK cells. So the presentation of MHC1 is essential for CD8 T cell recognition of cells, of antigen on tumor cells, but it is also actually an inhibitory signal for natural killer cells. Natural killer cells can recognize target cells, including tumor cells, through a different class of recognition molecules that recognize stress-induced ligands like MICA on tumor cells, but are inhibited by the expression of self-MHC1. So our hypothesis is that NK cells could be mediating this inhibitory effect. So we set out to test that by depleting different populations of immune effector cells in the KPC pancreatic cancer model, where we can show a clear sensitization effect of immunotherapy with JAK1 depletion. So when we deplete CD8 T cells, that effect is unchanged. CD8 T cells don't appear to be necessary for the sensitization of JAK1 tumors to immunotherapy. But when we deplete natural killer cells, the effect is completely lost, suggesting that NK cells are essential for this sensitizing effect of loss of function in the interferon pathway. Another way to test this is to see if NK cells are sufficient for the selection against interferon deficient tumor cells. So we can do that by isolating natural killer cells from the spleens of mice, activating them and expanding them in vitro, and then performing the same kind of competitive co-culture with tumor cells, except now in a dish where we have defined populations and we're mixing them in a certain ratio. So when we do that, we can see that when we add natural killer cells to a mixture of JAK1 and control tumor cells, we get a selective depletion of JAK1 guide RNAs with an increasing effector to target ratio, suggesting that the natural killer cells have a preference for killing the JAK1 deficient tumors. But importantly, this effect is lost when we delete TAP. So just like in our in vivo competition assay, it appears to be the case that the presentation of antigen is absolutely necessary for this effect, or when we delete the specific MHC1 heavy chain H2K1, which is known to inhibit natural killer cells. So this shows that NK cells are sufficient for the selective depletion of tumor cells that have loss of function mutation in the interferon pathway. So finally, we suspected that NK cells might be responsible for many of the MHC1 pathway and interferon pathway related hits from the screens. So to really hammer home this point, we performed an additional arm of one of our screens with immunotherapy and NK cell depletion simultaneously. And by plotting the NK cell depletion data against the original data, we could figure out which hits are specifically dependent on the presence of NK cells. So here in this scatter plot, I'm plotting the original screening data on the x-axis with depleted genes on the left and enriched genes on the right. And on the y-axis, I'm plotting the version of the screen we did with NK cell depletion. So genes falling on the x equals y axis here would be genes that are unaffected by the depletion of NK cells, but genes falling off the axis would be genes where NK cells were necessary for the observation of a dependency. So we, from this experiment, did conclude that NK cells are absolutely required for the observation that interferon deficiency is selected against in vivo. So we no longer see a sensitizing effect for loss of interferon gamma receptor, JAK1, JAK2, or STAT1 when NK cells are depleted. However, what was surprising to us is that many members of the MHC1 antigen processing and presentation pathway, including this non-classical MHC H2T23, were actually still very strongly depleted even in the absence of NK cells, and their hit strength wasn't really affected by NK cell presence or absence at all. The one exception was the classical MHC1 heavy chain H2K1, which I just showed a minute ago, had also suppressed the effect of NK cells selecting against interferon knockout tumor cells in a dish. So this was very mechanistically consistent with the data we produced in our individual competition assays, but this data was a surprise. So we set out to study this effect a little bit more because we hypothesized that H2T23 may be inhibiting a different population of immune effector cells. So before I get into that, a little bit of background about classical and non-classical MHC and a little bit about what HLA-E is and the nomenclature here. So in humans, classical MHC represents HLA-A, B, and C, and non-classical MHC heavy chains represent HLA-E, F, and G. And the difference between these two families is that the classical MHC1 heavy chains are highly polymorphic. They have a diverse epitope repertoire for the peptides they're presenting in the immune system, and they have broad expression in many tissues. And their major function is antigen presentation. However, non-classical MHCs are generally, they generally have low polymorphism. They present a limited epitope repertoire compared to the classicals, and they often have restricted expression, including to tissues like the placental trophoblast cells, and their major function is immunoregulation generally. And HLA-E is an important member of this non-classical MHC class 1 group that actually normally only presents a single antigen. So, and just a clarifying point on the nomenclature, because it gets a little confusing between mouse and human here. So in humans, both the gene and the protein are referred to as HLA-E. In mice, the gene is H2T23, and it encodes a protein that's called QA1B. So I'm going to refer to the mouse versions of the gene and protein for the rest of the talk. So what exactly does this inhibitory ligand HLA-E QA1 do? So this is the ligand for the NKG2A CD94 inhibitory receptor. And this is an inhibitory receptor complex that is expressed on both natural killer cells and T cells. And it's an important checkpoint, and actually anti-NKG2A monoclonal antibodies from Innate Pharma are currently being evaluated in phase 3 studies in combination with PD-1 blockade, and the preliminary data look quite encouraging from this combination. So remember that I mentioned previously in the talk that QA1 was actually the most common and important immune evasion mechanism across all of the screens that we conducted, and was recovered in 7 out of 8 of the genome-wide screens that we completed. And we know that deleting H2T23 sensitizes a wide range of mouse tumor models to immunotherapy. So here are 3 examples of B16 melanoma, this KPC pancreatic cancer, and a renal cell carcinoma model, all of which are rendered very sensitive to PD-1 blockade by the deletion of H2T23. And actually, when we looked back at our expression data, it occurred to us why we had only recovered H2T23 as a hit in 7 out of 8 of the screens we did. And that's because the only cell line where we didn't recover it, MC38, actually doesn't express H2T23 in either the resting state or after stimulation with interferon gamma, making it an outlier among the tumor models that we studied. So we validated this at the protein level. Here you can see that in these BRAF-B10 melanoma cells or in these pancreatic cancer cells, stimulation with interferon gamma upregulates the cell surface expression of QA1, but that does not happen in the MC38 colon carcinoma model. So we set out to determine whether or not the reason for not seeing this, this hit in MC38 was entirely related to expression. Is HLA-E QA1 expression sufficient to inhibit in anti-tumor immunity in MC38? So to do that, we actually engineered MC38s to express either a constitutive or an interferon-inducible version of H2T23 using this vector we made that has interferon-stimulated response elements and gamma-activated sequences upstream of the IRF1 promoter. So we showed that using these vectors, we could generate stable constitutive expression of QA1 or interferon-induced expression of QA1 in MC38s. And when we implant those MC38s into mice and treat them with immunotherapy, as in contrast to the control gene expressing MC38s, which we engineered to just express CD19, which are very sensitive to immunotherapy. You can see 75% of them are cured by the treatment that we're giving them. The QA1 overexpressing MC38s are very resistant to immunotherapy, and none of them are cured. So, we thought this was very interesting and important because MC38 is one of the most commonly studied models for anti-tumor immunity and response to immunotherapy drugs. And it actually lacks one of the most important immune inhibitory mechanisms that we discover across all of the in vivo screens that we've done. And when you give it back that immune inhibitory mechanism, it actually becomes very refractory to immunotherapy. So it's a bit of an outlier among the models. So a few minutes ago, I showed that from the depletion screen that we had done, it appeared to be the case that NK cells were not required for the sensitizing effect of loss of QA1 H2T23. We wanted to validate that using depletion studies. So we performed experiments with the KPC pancreatic cancer model where we deleted H2T23 or gave them a control guide, and we treated them with PD-1 blockade. You can see here that makes the KPC model very, very sensitive to immunotherapy. And when we deplete NK cells, we don't really have a strong effect on that particular that particular HLA-E sensitizing effect. You can see that in general, the tumors appear to grow a little bit faster here, but they're still very sensitive to the loss of H2T23. But when we deplete CD8 T cells, we completely lose that sensitizing effect, suggesting that CD8 T cells are absolutely required for the sensitizing effect of HLA-E deletion. So we wanted to understand what populations of CD8 T cells the cognate receptors, CD94 and NKG2A, are actually expressed on. So for that, we turn to our single cell RNA sequencing data. And you can see here, we have data from both untreated and immunotherapy treated KPC tumors. And we have T cells that fall into a few different clusters that we would expect to see. We have naive and these early progenitor CD8 T cells, which are mostly present in the untreated mice. And then in the immune checkpoint blockade treated mice, they differentiate into effectors, more of an exhausted phenotype CD8 T cell, and these proliferative T cells. And when we look at the expression of key genes that define the populations, like L-selectin or PD-1, we find them in the less activated or more terminally effector CD8 T cell subsets, as we would expect. You can see that the expression of cytotoxic molecules like perforin and granzyme B is higher in these effector CD8 T cells, suggesting that they are the populations primarily responsible for killing tumor cells. And when we look for the expression of the receptors, KLRC1 is NKG2A and KLRD1 is CD94, their expression is highest on these activated effector and exhausted CD8 T cells. So our hypothesis here is that interferon gamma is also having an inhibitory effect on CD8 T cells via the upregulation of QA1 HLA-E. But experimentally, that could be difficult to test and observe because the recognition of tumor by CD8 T cells also requires classical MHC1, which is interferon induced just like HLA-E is. So to circumvent this problem and actually test for the interferon regulated inhibitory effect that we're looking for, we actually turn to using a CAR T cell model to give T cells the ability to recognize tumor cells in an MHC1 independent way that wouldn't be responsive to interferon stimulation. So using this method, we could specifically try to determine whether interferon sensing is having an inhibitory effect on CD8 T cells, and then test whether HLA-E QA1 is responsible for that effect. What we would expect to see is that deletion of interferon would mimic QA1 deficiency and would result in a sensitizing effect to CAR T cell killing. So to do that, we set up an in vitro assay first, where we would take T cells out, transduce them with a CAR after CD3 and 28 stimulation, and then keep them in culture, and then co-culture them with KPC pancreatic cancer cells in a 50-50 mix that are H2T23 deficient or control. We also added IL-12 to the conditions here because we found that NKG2A is actually not expressed very well with the standard CD328 IL-2 stimulation, but adding IL-12 to the in vitro culture conditions substantially increases the expression of NKG2A, which would obviously be necessary for observing an inhibitory effect from HLA-E. So doing exactly that competitive assay, we confirmed that HLA-E is an important inhibitory ligand that specifically and directly inhibits CD8 T cells in an in vitro killing assay using this CAR T cell model. So you can see that there's selective killing of the HLA-E H2T23 knockout tumors. But the next step was to actually determine whether interferon is having an inhibitory effect on the T cells and whether that is controlled by QA1. So we're going to now perform the same competition assay, except we're comparing JAK1 deficient tumors to control tumors. And we're going to do that on either a control or QA1 knockout background, with the idea that in a JAK1 knockout tumor cell that can't sense interferon, that would mimic QA1 deficiency because interferon is required to upregulate QA1 on the tumor cells. So when we do that competition, we see that in the control condition, there is actually no preference for T cells killing either JAK1 or control tumors. This is actually in contrast to the data that's been generated by other groups. Remember at the beginning, I showed data from Jason Moffat's group showing that JAK1 deficiency made tumor cells resistant to T cell killing. If you use a CAR model, then that is no longer the case. Because PD-L1 is also an interferon-induced inhibitory ligand and the PD-1 receptor is expressed on T cells, we check to see whether anti-PD-1 would increase the selective killing of the control condition, which would implicate PD-L1 as the important immune-regulated inhibitory ligand. But that didn't have a very strong effect on this particular competition. However, when we delete QA1 on the control cells, you can see that now the T cells have an incredibly strong preference for killing the controls relative to the JAK1 knockouts. And this is mimicked by adding anti-NKG2A blocking antibody to the culture, suggesting that the T cells have a preference for killing cells that lack the QA1 NKG2A inhibitory interaction. And finally, we can set up an in vivo version of this exact experiment to ask whether in vivo HLA-E is specifically inhibiting T cells and whether that is the mechanism responsible for the interferon-regulated immune inhibitory effect on CD8 T cells. So we're going to use this exact same CAR T cell competitive killing setup, and we implanted into NSG mice a competitive mixture of control guide and JAK1 guide tumors. And then we're transferring in either untransduced or CAR transduced T cells, and then reading out the competitive assay. And we're going to do this again on the background of either a control tumor or tumors that lack expression of QA1. And when we do that, we see that in the setting where the mice are given T cells that are specific for the tumor cells, we engineered them to express CD19. There's a strong preference for killing now the JAK1 knockout tumors in vivo, but that is reversed almost completely when we do the experiment on the background of QA1 deficient tumors, suggesting that interferon is having a specific inhibitory effect on CD8 T cell killing in vivo, and that effect is predominantly mediated by the upregulation of QA1. So the model that we've developed from doing this work is that when immunotherapy is used, you have both activated CD8 T cells and NK cells that begin to recognize tumors through a variety of different mechanisms. MHC1 for the T cells, and probably ligands for NK cells like NKG2D ligands and other ligands recognized by other NK cell receptors. And both of those populations produce interferon gamma. The tumors sense the interferon gamma and in response upregulate MHC1 heavy chains, both classical and non-classical MHC1, to a very high level. And the upregulation of those MHC1 molecules has an inhibitory effect on each population, with classical MHC1 and to some degree non-classical MHC1 having an inhibitory effect on NK cells, and especially non-classical MHC1 like QA1 having an inhibitory effect on T cells. So in the screens, what we're seeing is that loss of the interferon pathway by either ablating the interferon gamma receptor or the downstream molecules necessary for sensing and signaling interferon are blocking that induction of MHC1 heavy chains and allowing the immune effector cells to kill the tumor cells more effectively. That is, if there's at least some expression of MHC1 that allows CD8 T cells to still recognize tumors. So in conclusion, what I've shown today is that in vivo genetic screens can enable the systematic discovery of novel mechanisms of sensitivity to immunotherapy, and that HLA-E, QA1, and interferon sensing arise as the major mechanisms of immune inhibition in vivo. And we've uncovered that interferon actually inhibits both T and NK cell functions through classical and non-classical MHC upregulation, and that NKG2A is a critical checkpoint that regulates CD8 T cell cytotoxicity. So I want to acknowledge all the folks in my lab that helped to do this work. It's been a huge project that we've undertaken over the course of many years. Everything that I've done was done in close collaboration with Kathleen Yates, who runs the entire TIDE operation at the Broad with me. And Juan Dubrow and Peter Du were the people leading the TIDE screening and analysis effort from the experimental and computational side. And they've been working with a dedicated team of technicians that include Sarah Kate Lang-Riddicker and Audrey Moscato, and many others who have helped make these really large and complex experiments possible. So thanks to them and to all our other collaborators, and I'd be happy to take any questions.
Thanks very much, Rob. This was a great talk. We just have about under 5 minutes for questions, so I'll read them quickly and ask for responses as I go through them. The first question here is from Ling Kai, who's curious about the HLA-E results, particularly because in humans, there seems to be a positive correlation with response to immune checkpoint inhibitors. So he's asking whether you've tested humanized mice, or what are your thoughts about human-mouse differences?
Yeah, I can answer that one quickly. I think HLA-E is probably also an important inhibitory mechanism in humans, and I think some of the NKG2A blockade clinical data is bearing that out. I think the response, the association with response to immunotherapy is probably because HLA-E is highly induced by interferon gamma, and interferon gamma signatures enrich for patients who are likely to respond to immunotherapy in many tumor types like melanoma.
Thanks, Rob. Next, we have a question from Rakesh Jain, who's asking about differences between subcutaneous versus orthotopic microenvironments and whether that would affect your results.
Yeah, that's a great question, Rakesh. We haven't done yet screens with tumors in the orthotopic location for many of these different models, but we have done some validation studies of individual hits in different tumor locations. And I'll say that largely, at least for the story that I've told here today, the validation results are very similar. You know, I think the one exception could be if there are particular tissues where NK cell effector function is impaired, you might not see these results in the same way. But for any tissue where you'd expect these immune effector cells to be present, I would think that this biology would largely be conserved.
We have a question from Hiroshi Egawa about whether the CRISPR screening data will eventually be available on DepMap, Portal, or other web tools.
Yeah, great question, which I can answer affirmatively with. There's a computational biologist in our group who's actually been working very hard to build the data access portal for all the data that I've showed here today. And when our paper is published, we're going to make that portal publicly accessible. So it will be available, and the raw data will be available with our publication, but there will be a browser for the community so that you can quickly look up individual results in many of our screens.
And another questions on your hits. You've compared primarily the ICB, the immune checkpoint treated mice, versus NSGs. Do you also see the same differences if you compare just wild type mice who have not been immune challenged?
Yes, there are some differences between the wild type animals and the spontaneous immunity that we observe in those groups and the animals treated with immune checkpoint blockade. Largely, many of the same pathways are recovered, but there's definitely a difference in the relative effect size. In the animals treated with immunotherapy, we see a greater effect size for the depletion and enrichment of most of the same pathways. And there are some individual differences in the recovery of specific hits. For instance, although HLA-E is observed as a hit in both cases, it's actually stronger in the immunotherapy treated cohort compared to the immunodeficient mice.
There's another question from Patrick Anderson about a recent paper describing CRISPR-induced structural variants and whether you think that that could also contribute to the immune response.
I do not think that CRISPR cutting induced structural variants are inducing an immune response in our models. And I think our experimental system is quite well controlled for that effect.
And one other question is in terms of saturation of your screens, you have heterogeneity among different tumor types and different cancer types. Do you feel that you achieved saturation for each one and therefore they're really not overlapping between different cancer cell line models? Or do you think that ultimately you would be able to cover the same hits and have more overlap among your 8 different tumor models?
If the question is, if we did more screening and screened with more mice or more guide RNAs, would we eventually recover the exact same hits? I think the answer is no. I don't think they would begin to converge more with more screening data. I think that there are some things that are clearly shared, like the interferon pathway and HLA-E that I've talked about today, and those things, we recover quite strongly in all of the screening data that we've generated. We don't need more data to see those effects. And there are a lot of things that appear to be specific to the models, and that's not necessarily because they just have marginal effect sizes and more screening data would recover them in all the models. It appears to be the case that they really don't cause immunotherapy sensitization in one cancer cell line, but they do, and in some cases, we've validated that they do in another.
And now last question that we have time for is from Mengling Wang. Is there a difference between tumor cell and normal cell and expression of QA-2?
Of QA2 or do you mean QA1?
It says QA2, but let's make it QA1.
Yeah, we don't know too much about QA2, and that doesn't have a super clear human homologue that has exactly the same conserved functions. So I'll answer for QA1. QA1 is an interferon-induced gene, and it does have expression after interferon stimulation on many on many tissues. We think that it's unlikely to have a significant effect on immunity in many other tissues besides the tumor site. And one of the reasons I think that is because there's a knockout mouse of QA1, and that mouse is largely healthy. It has phenotypes only in response to a few different infection challenges and autoimmune disease challenges. And also the emerging clinical data from the NKG2A blockade antibodies appears to indicate that it is safe.
Thanks, Rob. So for everybody whose questions we could not get to, they will be sent to Rob and he'll have the chance to answer them by email. And then Claudia from DFACC will be sending them back to you so you'll have your questions answered. So thank you very much, Rob, for a fantastic seminar. Thank you everyone for attending and have a wonderful day, everyone.
Thank you, Daniel, and thank you everyone for listening to my talk. Have a good day.